Chapter 15 deals with the microbial mechanisms of pathogenicity. In other words, how is it able to infect you? Once again, we've got different definitions here for various terms. Infection just means that the pathogen has entered the body and it has multiplied. You may not know it's there yet, but it is. Disease is now when you have an abnormal function that's caused because of the pathogen being there. So you can have an infection without having disease. Like I said, you may not even know that the pathogen is in the body. Signs and symptoms. We use the term sign, meaning it's something that's subjective, something that can be measured. It can be observed by somebody else. If you have a temperature, you can measure that and see, oh, there's a fever here. There's, it's 100 degrees, or it's 102, or whatever it is. A symptom is something that's subjective. It's felt by the patient, but you don't have a measurement for it. Oh, I feel fatigued. How do you put a measurement on fatigue? You can't. Asymptomatic means that you have the disease, but you're not showing any signs or symptoms of it. So once again, you probably don't know that you have it. For signs and symptoms, here are some examples. Signs, like I say, temperature, blood pressure, heart rate, breathing rate, antibody titer. You can measure all of those things. Symptoms, nausea, loss of appetite, pain, fatigue, aches. You can't put an exact measurement on that. You just have to go off of what the patient is describing for it. It's important to know what the signs and symptoms are, and being in healthcare where you're dealing with the patient is to make note as they're describing how they're feeling, whether they do feel nauseated. Okay, you can't measure that, but you need to mark that down that they're feeling nauseated. If you can measure, say, a fever, yes, measure the temperature, write that down. Can you diagnose a disease purely from the signs and symptoms? No. Because a lot of them have the same things. Stop and think about it. How many things do you have where symptoms might be fatigue? You're tired. You're kind of achy. Just fatigue alone. Well, that pretty much describes every single college student I know. Just kind of aches and pains. Think about in a little bit more detailed terms. Runny nose, fever, aches. That describes just about every single infection that is possible. So they're all clues to help you with the identification of what the infection is, but you can't use that solely by itself. Classification of diseases. So if it's an infectious disease, it's due to the pathogen, whether it's bacteria, virus, fungus, whatever. If we say it's communicable, that means it can be transmitted between individuals directly or indirectly. If it's contagious, it's very easily transmitted between individuals. You walk in a small room and somebody has contagious disease, you're probably going to get it. Non-infectious, this means the disease typically is going to be due to genetics and or the environment. Non-communicable, it cannot be transmitted from one person to another. A nosocomial infection, this they used to say was hospital acquired. We've now changed that to healthcare setting acquired, meaning it's not just hospital, but maybe at a nursing home, a care facility like that. And typically what happens is a person comes in like to a hospital with one infection and they may pick up a nosocomial. They pick up the secondary infection. Zoonoses are diseases that are transmitted from animals to humans. Acute means short-term, chronic is long-term, latent means that it's dormant. So we talk about a lot of these different ones as we discuss them. Oftentimes what we are doing for diseases, you will take samples, oftentimes a blood sample, maybe a urine sample, and you're going to be looking at the sample. You know what normal looks like, which is why we always stress in classes such as A&P. You know what the normal looks like, so now you can see what the abnormal is like. Over here, the slide on the left side, excuse me, You're looking at a blood cell line. Most of these are red blood cells that you're looking at. And then you see what is labeled here. Sorry, as the, oh, and this will not go back. I'm sorry. It's just showing a Plasmodium, which is causative agent of malaria, versus, in the other slide, kind of looks somewhat similar, but it was sickle cell. That's a genetic disease. So you're talking about infectious versus genetic there. What are some types of non-infectious diseases? You have an inherited disease. You can have congenital that you're born with, degenerative, nutritional deficiency, endocrine. Endocrine refers to hormones for those of you who have not had A&P. Neoplastic or idiopathic. Idiopathic is one where we don't know what the cause of it is. When we look at diseases, we talk about the stages. There's 5 distinct stages of the disease. There is an incubation period, the prodromal period, the period of illness, period of decline, and finally the convalescent period. If you graph this out, this is what you see. And so let me get my little marker here. This first period, which is the incubation period, this is when you have been exposed to the pathogen. It has infected you. It is starting to replicate inside of you, so you have the infection. You may not know it at this point. You may be perfectly fine. Maybe you came to class on campus in the morning. You were perfectly fine when you woke up, and you happened to meet somebody who maybe had a cold. You didn't think anything of it. They're sneezing near you. And this first incubation period is after you've seen that person. You're feeling okay. But if you look at the, over on the left-hand side, this is the number of organisms, pathogens that are in you. It's starting to increase. Prodromal period, this is where now you're starting to go, yeah, you know what? I have a little bit of a runny nose. Well, is it allergies, or maybe I'm starting to pick up a little something? I don't remember having a runny nose when I got up this morning, and maybe you're starting to feel a little achy and you're going, well, I don't know if I'm coming down with something, or, you know, I've been up a lot, really late lately, just putting so much time in studying for this microbiology course. I'm not getting as much sleep anymore, and so I'm just tired. Maybe it's just because, I mean, I am a college student, and so I know I'm not getting sleep. I'm not eating right. I know you're not supposed to eat pizza for breakfast, lunch, and dinner, but hey, that's what I eat right now. Period of illness, now you know you are sick. The number of pathogens has greatly increased to the point where you know you're sick. You know this is not just a case of you haven't gotten enough sleep lately and you're not eating well. It's, I don't feel good. I apparently was exposed to somebody else who was sick, and I picked up something because now I have a fever. I ache all over. My nose is running. You may or may not feel nauseated. I don't feel good. This will continue on. And then finally hit the point where you're like, okay, now I'm coming through the period of decline here. The number of pathogens is decreasing, where you're going, okay, I'm starting to feel a little bit better. I am still not okay. This is where some students are like, I'm feeling better, but I don't feel like coming to class. I don't want to listen to her lecture. I'm just so tired. I just don't feel good. You're improving. You know you're going to make it. Period of convalescence. This is the final period where, yes, you're still sick, but you're improving. The number of pathogens is decreasing. This is a period where a lot of times this can be a problem for individuals where they know they're not completely well yet, but they start pushing it. Oh, I'm well enough to go back to work. Oh, I'm well enough to go do this. I have so many things I have to do. My family needs me. My coworkers need me. I need to get this stuff done. And you start to push a little too much too soon. So the period of convalescence is kind of a danger zone of don't do too much too soon, because you don't want to push it to the point where you're going to get sick again. You're going to pick up something else. You're going to make that period of convalescence actually even longer than it should have been because you tried to do too much too soon on there. How long do each of these sections here? You notice it's just this time here. It depends on the individual pathogen and the disease as to how much time a person will spend in each of these 5 different phases. It may be in hours, it may be in days. It just depends. Koch's postulates, Robert Koch developed these 4 rules or 4 steps of how do you determine what is the causative agent of a disease. And so this is what we do. We follow these 4 steps even today. Number 1, the suspected pathogen must be found in every case of the disease, but it is not found in healthy individuals. That's step 1. Step 2, you must take that suspected pathogen. You must be able to isolate it and grow it in pure culture. Then what happens is you take that pathogen that you grew in the pure culture from step 2, you take that pathogen and you infect a healthy individual. And you see if they develop the same signs and symptoms of the disease that everybody in step 1 had. And then from that person that you infected, you must be able to isolate the pathogen from it, and it must be identical to the pathogen that you found from the individual in step 1. Those are the traditional Koch's postulate steps. Now, in a perfect world, yeah, this would be great. However, we cannot always follow these. One of the problems is right here in terms of being able to grow it in a pure culture. We are not able to grow everything in pure cultures. Bacteria do not come with an instruction manual saying, hey, this is what I need. One example of this is the bacteria that causes leprosy. To this day, we cannot grow that. It's a bacteria. We cannot grow it in pure culture. The only place we can grow it is in the footpads of white mice and in armadillos. That's it. However, at this point, we have found that this bacteria is in every single individual that has leprosy, and it is not seen anywhere else. So we're able to pretty conclusively say, okay, this bacteria is the causative agent. The other problem that we have, and this involves ethics, and that is with this 3rd step here of where you are going to infect a healthy individual. If the disease is fatal and there is no cure for it, you cannot ethically infect a healthy individual with a pathogen that is known to kill you. You can't do that. That is morally and ethically wrong, and so that is not allowed to do. Has it been done in the past? Unfortunately, yes, sometimes it has. And thank goodness we have enough oversight and governing within the scientific community itself, as well as government agencies, to say, no, there are rules in certain steps that must be adhered to for safety. Safety of the individuals involved with the testing, safety of everyone involved. So there are times when, once again, you may find a pathogen and you find it in every person who has a particular disease, such as like HIV. We have found that virus in every person who has HIV. It's not present in people who don't have HIV. Can you take that virus? Have we been able to grow it in the lab? Yes, so we can work with it. So we've been able to check off 1 and 2 as far as the steps of Koch's postulates. When there is no known cure for HIV, same thing like Ebola virus, there is no known cure for that. Can you infect that into a healthy individual just to see, are you going to come down with HIV, or are you going to come down with Ebola? No, you cannot do that. So ideally you would cover all 4 of these steps of Koch's postulates, but in reality we understand that sometimes you can't do that. You just cover the ones that you can, And then, like I say, if you're always finding this virus with this disease, then you can say, yes, this virus is the one that's causing Ebola or HIV or whichever one you were talking about. So sometimes we have to kind of amend postulates with that. And that's what this is just showing, that you have over here, excuse me, your diseased animal, you have your healthy animal on the top here. You take your cultures, you look at it, nothing's there. On the diseased animals, you look at your cultures, here's the suspected agent growing on a plate. Here's a healthy animal, you inject your culture into it, and then you're able to this starts to show the same disease, and then you isolate the same organism. Ideally, that's the way it works. Pathogenicity refers to the ability of a microorganism to cause a disease. Virulence is what is the degree to which the pathogen can cause the disease. If something is highly virulent, yeah, it probably is going to cause the disease. If it's a low virulence, okay, the possibility is there, but probably not. Avirulent means it doesn't cause the disease at all. Median infectious dose, refer to this as ID50. This is the number of pathogenic cells that are required to cause an active infection in 50% of inoculated animals. This is going to vary depending on the species. There's a bacteria, Shigella, a causative agent of several different things, including dysentery. You only need 10 to cause. The LD50 is 10 cells. That's it. That's not very many. Median lethal dose, the LD50, this is the number of pathogenic cells that are required to kill 50% of the infected animals. So are you looking at what's going to cause an infection versus how many are going to kill the host? And you can graph this out. This is usually what they will do is do experiments, testing various numbers of the pathogenic causative agent, and then see what is the LD50, what is the ID50. Sometimes you hear about a primary pathogen. This will cause a disease in any healthy individual. Okay, it's a true pathogen. Sometimes you'll see that term. Anyone you're exposed to it, you're going to get sick. I don't care what your medical background is, it's going to get you. An opportunistic pathogen, as the name implies, this will cause a disease in somebody who's immunocompromised. Maybe they have another illness, maybe they're being treated, say, chemotherapy for cancer. Their immune system is already suppressed. It's stressed out or something else. So it's kind of like opening the door, making you more susceptible. So an opportunistic pathogen on a healthy individual probably wouldn't make him sick, but on somebody whose immune system is compromised, yeah, it's going to make him sick. Sometimes what it is an microorganism, bacteria, or whatever, that is found in part of the normal microbiota, meaning it's normally on you or in you, but now it's in a different location. You move it to a different location, and now it's going to cause a problem. So secondary infections, this one, your body's already got an infection and it's responding. The immune system is trying to fight it. We've got invaders and we're overwhelmed because we're trying to fight this infection. And now there's something else that comes in. So you've kind of been overwhelmed and focused on one area trying to fight that, and then something's trying to kind of come in the back door without you knowing it. And next thing you know, now you have a second infection. So how do microorganisms enter? We refer to it as the portal of entry. This is the location of where the pathogen enters the body. Oftentimes it can't get in. Now we're going to look at how the body responds to things trying to enter that aren't supposed to be there. What we are looking at right now is how does the microorganism enter in the first place. And later we will talk about how it leaves. That's known as the portal of exit. Wherever there is an opening to the external environment. That's kind of like a window or a door. Provides an opportunity for microorganisms to enter. So through the eye, and some people say, well, the eye, the eye socket, the optic nerve goes through the back, enters the brain, through the pupil. Remember the pupil, that black circle in the middle of the eye? That's actually a hole in the eye. It's allowing the light in. Well, if it's a hole, that's an entrance. There's a reason why you have the cornea in front of it. There's a reason why you have your tears that are constantly flushing over the eyes. Your nose, your mouth, you're inhaling things, you're ingesting things. Ears, things can enter through the ears. If it gets in the ear and it's able to penetrate and get past the tympanic membrane, which is the eardrum, it's then in the middle ear, which has a eustachian tube attached to, which drains down to your throat. The urinary tract, your gastrointestinal tract, from the mouth down through whatever you eat, the stomach. As my father likes to say, over the teeth, over the gums, look out, stomach, here it comes. It's going from the mouth down, the pharynx, esophagus, to the stomach, to the intestines, and then the genital tract. The most common portals of entry are with the respiratory and the gastrointestinal tracts. So inhaling and eating. So this picture is just showing some of the different portals of entry that are common. Now, on the skin is very good at protecting. It's a barrier, or physical barrier, as we'll talk about later. But if you have a scratch, you have a break, a tear, then it is able to enter in through that. Stages of pathogenesis. When the pathogen enters, exposure, that's when it enters the body. It's like, whoa, I'm in. Adhesion is when it sticks or adheres to the cells at the portal of entry. If something comes in, let's say through the nose, okay, it got in the nasal cavity, and then if you sneeze, if you're like my brothers, I'll just say when my brothers sneeze, you can hear it probably next door. They don't know what a quiet sneeze is. It is huge. And so we'll just say we're at the hospital recently with my dad. My brother started sneezing. He was out in the hallway. A nurse came out of a room 5 rooms down the hall to see what was going on. That's how loud it is. So for a bacteria to get in the nasal cavity like that, and if you sneeze like that, okay, it's out of the body. The only chance it possibly has to stay in the body is if it can stick to some of those cells there at the beginning. So just getting in is not going to help you. You have to adhere. You have to stick to those cells. And then you're going to invade. You're going to move throughout the body. If you got in through the nose, and maybe you want to get down to the lungs. So you got to start your track. You got to start moving down there. Infection is when now it starts to replicate. So you got in with one cell, replication in a short time, as you guys know, you are greatly increasing the number of cells there. Transmission, this refers to when the pathogen is going to leave that infected host. So if it infected you, it leaves you by the portal of exit, and it's going to move on to somebody new. No offense, but we want to keep infecting somebody else because that's the way it's going to stay alive. So in this picture, it is showing that the bacteria are able to produce, remember the glycocalyx? That's extracellular material, and part of what it is doing is helping it to stick to a surface. This is a catheter, and you can see all these little circles here are the individual bacteria. But they have these little things sticking off from it. It looks like a sticky, gooey mess. And that's exactly what it is. It's helping it to stick to the tubing. H. pylori, which is the causative agent of stomach ulcers, it's able to invade the body. It's able to survive in the stomach, even though it's very, very acidic there. It starts producing certain, what we call virulence factors, that help it to pass through this layer right here. which is made to protect the cells of the stomach. And what it's going to do is it's going to penetrate through there until it reaches the cells underneath. But now these cells underneath, right here, it's exposed now. The stomach acid is, so this is inside the stomach here. It's now exposed. So when pathogens leave the body, like I said, they leave by portals of exit. Oftentimes they can be the same as the portal of entry. So how do they leave? It has to be somewhere where there's an opening. So it can be the nose, the mouth, the eye, the ears. It can be through the genital urinary tract. It can, if you have a break in the skin, it might leave through there as well. So virulence factors, these are factors that the pathogen has. In most cases we're talking about bacteria, but it can be viruses or fungi as well. So it's what the pathogen has. Virulence factors, it's going to help make, the more factors they have, the more virulent the pathogen will be, meaning it increases the degree of pathogenicity, the ability to make the disease. The more chance it's going to basically make you sick. So there's different classes of them. There are several different, what we call exoenzymes. These are enzymes that it makes and it exports. Things like hyaluronidase, which is helping to hold cells together. DNase breaks down DNA. Collagenase. If some of you I know have had AMP, some of you may not have had AMP. Collagen is very important. It's part of the extracellular fibers that are found in connective tissue. And so it's helping to hold the tissue together. Well, here comes a bacteria that produces collagenase, and it breaks it down. And if it's also able to make hyaluronic acid is between also the cells, helping to stabilize and hold them together. And so if a bacteria is able to produce both of these enzymes, it allows it to penetrate from the surface of the skin. It's able to penetrate deeper into the body. Haha, I just got in. Toxins. There are different types of toxins. There's endotoxins such as lipid A that is made by gram-negative bacteria, and then several bacteria make it exotoxin. You also have capsules and adhesions. So if we look at these, as I said, hyaluronic acid, this compound here that's helping to hold adjacent cells together. And if the bacteria comes in and it produces hyaluronidase, guess what it is? It's breaking down this connection that's holding these cells together. So if you're on the outside here, guess what? You produced that and you just got inside. You just penetrated through part of the defenses. Similar thing with the collagenase, where you have collagen fibers that are running through here and helping to hold the tissue together. You just got in because you broke it all down. Remember that gram-negative bacteria have a very thin layer of peptidoglycan, and then outside of that, so on the surface of the cell wall, is a lipopolysaccharide membrane. This lipopolysaccharide membrane has lipid A. It's on the end of it right here. Lipid A triggers the inflammatory response. This, if you remember, when we were talking about the cell wall and the chemical composition and structure of it, I had mentioned then that if you have a gram-negative bacteria and you have an infection with it, you need to be careful. You do not want to kill them off all at once. And the reason for that is because you don't want to dump all this lipid A. It will trigger that inflammatory response, and if left unchecked, it can put your patient into shock, and it could actually end up killing them. So that's a defensive mechanism, if you will, of the gram-negatives. Like, try to start killing me, and if you don't kill all of us, but you kill some of us, we're going to dump out this lipid A, and that's going to make you really sick. And hopefully then you stop trying to kill us. Toxins. two big toxins that we will talk about when we talk about diseases. You've got the botulism toxin, which is here, and the tetanus toxin, which is down here. They are both produced by bacteria. They're actually the same genus but different species. They're both commonly found in the soil. They both interfere at the neuromuscular junction, but in opposite ways. The botulism toxin, if this toxin is released, what happens is, and I know for some of you who have had AMP at the neuromuscular junction and how muscle contraction works, it's maybe bringing back nightmares, hopefully not. Under normal conditions, you have the neurotransmitter acetylcholine. And what happens is you have, I'm trying to find it, there we go. You have your action potential, the electrical impulse coming down the nerve under normal conditions. It is going to essentially trigger the release of the neurotransmitter, which is acetylcholine, from these vesicles. It's going to move across the synaptic cleft, across the space between the neuron and the muscle, and it's going to bind with the muscle, open the gates, and trigger the muscle contraction. What happens with the botulism toxin is you would still have your action potential coming down here, but the toxin prevents the release of that neurotransmitter. So acetylcholine is never released to come down and bind. You therefore never get muscle contraction. It is a form of paralysis because you never get muscle contraction. That obviously is a huge problem. Now we will talk more about this when we talk about diseases and exactly more properties of the toxin and how it works. And at that point, yes, is this the same toxin that's used for Botox treatments? They use a diluted form of it for that. Just be careful with it. Because in case you don't know, botulism toxin, that is the most toxic natural compound known to man. So I'm just going to throw that out there. Tetanus toxin. What this does, the bacteria, it's a similar way. It's going to release the toxin. Now, normally what happens when you have the action potential come down, you release, just like up here, you release the acetylcholine, that neurotransmitter. It triggers the muscle contraction, but you have enzymes coming over here that will break down to stop the contraction. They break down the acetylcholine, reaction stops. With the tetanus toxin, what happens is you still have your charge coming down, but you don't release the enzyme that's going to break down the acetylcholine. So all of this acetylcholine that's here is never taken away. And so you have a continuous contraction. You never stop the contraction. Either way, both of these are bad situations for the patient. Producing toxins is a virulence factor. It's an advantage for the bacteria. Capsules. I believe I mentioned this before. If an organism has a capsule, it makes it easier for protection for the bacteria. It makes it harder for our cells whose whole purpose is to search out and destroy foreign things by eating it. It's harder for them to eat because it's bigger. They can't get it. This picture on the far left is showing a micrograph. You're looking at cells. In this particular one, you've got two cells, typically the way they look, and then this clear whitish space in here. This is a capsule. This one right here does not have a capsule around it. So it's more susceptible to being eaten by the defensive cells of our system. So that is why we say if it has a capsule, that's a virulence factor. It's protection. If it does not have the capsule, and we'll use the term non-encapsulated bacteria, it's more susceptible to being broken down, so therefore it has a very low virulence factor. Antigenic variation in viruses. This helps to protect the virus in this case. An example of this, classic example, is the influenza virus or the flu virus. It has what's known as antigenic drift and antigenic shift. With the flu virus, there are proteins that are known as spike proteins, and they're given the letters H and N. If there is a point mutation which causes just a slight change in those proteins, that's known as antigenic drift. And this occurs fairly frequently. It's just a small change, small mutation in it. Antigenic shift is when there's a major change in those spike proteins. And that's typically when we see a bad flu season, because it's more of a larger mutation, a bigger change in it. We give these proteins numbers. So just so you know, for the flu, we do designate it influenza virus, but then we will say H and we give a number, and N and then give a number. So it might be H1N1 or H1N2, etc., as to how we designate it. But we talk about the shift in the drift. So shift is just a minor, small mutation, slight changes in those proteins, spike proteins. A shift is a major change in those spike proteins. And so this is just showing on here the proteins, and as you can see, we just abbreviate as N and H, and we give them the numbers for them. Your eukaryotic pathogens, your fungi, your protozoans, your helminths. Fungi, kind of similar to bacteria in that they will produce adhesins to help them stick once they're inside the body. Some of them produce toxins. We call them mycotoxins. Myco means fungi, so it specifies that it's a toxin produced by the fungus. And they may also produce some enzymes to help them penetrate deeper. Protozoans, they will sometimes release substances that are cytopathic, meaning that they will damage or kill our cells. They will produce sometimes adhesions to help them stick. Depending on the protozoan, some of them produce capsules, also provides protection for them, or some variations in their antigens, which makes it, the antigen is something the foreign substance makes, so it makes it harder for our immune system to detect it. Helminths, which are worms, they often have an exterior coat that has glycan molecules on it, basically mimicking our cells. Kind of, let me kind of look like you, so you won't realize till it's too late that I'm not you. And sometimes they work to help to suppress the immune system as well.